This is the phospholipid. There are two parts to the phospholipid, the phosphorus head and the fatty acid tails. They are bonded by the phosphate group. Because the phosphorus head is polar, it is hydrophilic, which means it loves water. The fatty acid region is nonpolar, which makes it hydrophobic. The fatty acid region hates water. The phosphorus heads face towards the water and the tails face away from the water.
This is a phospholipid bilayer. This is cholesterol. Cholesterol provides some stability to plasma membranes of animal cells. In high temperatures, it helps maintain firmness. In cold temperatures, it helps keep it flexible. This is a carrier protein. Carrier proteins attach to specific molecules, which then are transferred across the membrane. During this transfer, the carrier protein changes shape. This is a channel protein. Channel proteins provide open passageways through the membrane for certain substances to be able to pass through. Some substances are too big, but some fit just right to go through. Both of these are integral proteins and transport proteins. Integral proteins are proteins that go through the phospholipid bilayer. Transport proteins are proteins that transport things across the bilayer. This is a peripheral protein. Peripheral proteins are a type of protein that are loosely attached to the surface of the cell membrane, rather than being embedded within it. They provide structural support, and they act as receptors for extracellular signals, transmitting information into the cell. Some peripheral proteins also provide movement of molecules across the cell membrane.
So now let's go ahead and look at cell transport. First, we have the concentration gradient. The concentration gradient is a difference in how crowded molecules are in two areas. As we see in the diagram here, molecules tend to move from crowded areas or high concentration to less crowded areas, low concentration, until it is even. You can think of the concentration gradient as imagining yourself walking into a room where perfume has been sprayed. All the molecules are crowded on the side in which the perfume was sprayed. But as time passes, the molecules spread across the room, allowing all the people in the room to smell the perfume. Now, let's go ahead and look at selective permeability. Selective permeability means that the cell membrane is picky, only allowing certain molecules, like water or smaller molecules, to pass through, while disallowing big proteins or stuff that the cell doesn't want to not pass through. You can think of selective permeability as a bouncer in a nightclub, only allowing certain important people to pass through and not allowing other regular people to pass through. After that, we have our solutes and our three types of solutions. Our first one is isotonic, which means that there's the same concentration of solutes and solvent. You could think of this as the same amount of water as there is Kool-Aid. Next, we have our second solution, which is hypertonic, which means that there's more solutes and less solvent. You can think of this as there being more Kool-Aid than there is water. And lastly, we have our third type of solution, which is hypotonic, which means that there's more solvent and less solute, which you can think of as more water than there is Kool-Aid. Moving on now, we have passive transport with diffusion and facilitated diffusion. Passive transport is when molecules move across the cell membrane without using energy. You can think of this as them just going with the flow. They move from high concentration areas to low concentration areas, so you can think back to the concentration gradient. Simple diffusion is when molecules move freely through the membrane without any help. This is when oxygen or carbon dioxide slip through the cell membrane. Facilitated diffusion is when some molecules are too big or charged to sneak through the membrane on their own. Like simple diffusion, there's still no energy needed, but instead, proteins, either channel or carrier, to pass through. So now, second to last, we have active transport. Active transport uses energy, or ATP, to move things against the concentration gradient, moving from a low area of concentration to a high area of concentration, almost like pushing a boulder up a hill. In every cycle, the pump kicks out 3 sodium ions and brings in 2 potassium ions from the outside to the inside. In turn, this keeps the inside of the cell more negative and balanced. And lastly, we have bulk transport. Bulk transport is when the cell uses energy, or ATP, to move large amounts at once. Bulk transport comes in 2 types, endocytosis and exocytosis. Endocytosis is when the cell wraps its membrane around molecules and pulls it inside like a bubble. You could remember this with the prefix endo, which you can think of as entering. Exocytosis is when the cell packages molecules into little bubbles or vesicles and pushes them out. You can remember this using the prefix exo, which you can think of as exiting. And that was the cell membrane and cell transport. Thanks for watching.